Hi, my name is Barry Walsh, producer of the BPI conference. I'm here with Dr. Michael Butler from the University of Manitoba. Hello, Michael.
Hello, Barry.
So we're just going to talk today about the BPI event and ask Michael a few questions here. We're going to start with, what are you currently working on?
Well, the main thrust of activity in my lab is mammalian cell technology. So we are working on monoclonal antibodies as the primary target of our work, and particularly on glycosylation of monoclonal antibodies. We're very interested in knowing the structure and functional relationship of glycans, knowing that it's that function which is important when relating a monoclonal antibody to a therapy. So that's a very important theme of our lab at the moment.
And where do you see these technologies going over the next 5 to 10 years?
Well, I think the technologies are going to be connected with the production still of monoclonal antibodies, but particularly specifying targeted monoclonal antibodies. They may be, for example, conjugate antibodies. They may be small antibodies. We have some very interesting antibodies that come from camels and llamas, which are just 18 kilodaltons rather than the usual 150 kilodaltons from human. And they have, I think, a great potential of increased clinical use through being able to penetrate tissue. So it's that type of specialized antibody or an antibody with a specific glycan profile, which I think is going to be important in the future.
Great. And what do you see as some of the biggest challenges facing the bioprocessing field?
I think the biggest challenges, and I think that they're going to be important opportunities in biotechnology, are going to be the commercial aspects of biosimilars. Because I think biosimilars are going to increase the competition in the production of recombinant proteins and monoclonal antibodies, and hopefully bring drugs to the communities at lower cost and hopefully with increased functionality. So, you know, some of them may be considered bio-betters, and that's a very good, but that is going to be a commercial challenge from the point of view of the companies competing with one another.
What new technologies do you think are going to be emerging in the field?
Well, I spoke this morning about a very new area, certainly new to me, and it's new to most people, and that's dielectric spectroscopy. And we have found that we've been able to develop an instrument which is capable of looking at subpopulations of cells. In our case, the case study was based upon apoptosis. So we were able to identify subpopulations of early, mid, and late apoptotic cells. And I think that this type of non-invasive technology is very important, because there's a basis for being able to look at the electrical properties of cells and determining some of their capabilities and some of their metabolism. And I think that's a growing area in the field.
You've been with us since the very beginning. What do you consider your favorite aspects of the BPI conference?
Well, the BPI conference has grown in terms of the number of representatives, delegates here, and it's an excellent opportunity for networking. And for myself, coming from academia, working in bioprocessing, it's very important to know what the industry trends are, what are the limiting steps in the bioprocess. And so it's by this type of interaction that it makes the whole meeting very, very interesting.
Well, thank you, Michael. We really appreciate you being a part of this event for so many years. We hope you enjoy the rest of your week and your time here in Boston.